Welcome everyone to week number 4 in Biology 120 Lab. We're going to be doing an experiment this week to look at cellular respiration and how that happens and how much occurs over time. So this is a study of metabolism. Metabolism, the basic definition of that is all the cellular biochemistry that's happening. So all the chemistry that takes place, breaking down food, making new molecules that you need. That's it. That's what metabolism is. So last week we learned about diffusion, which is the movement of molecules from areas of high concentration to lower concentration until equilibrium is reached, and that is just a law of physics out there that molecules do this. They do it faster when the temperature is higher. They do it slower when the temperature is colder. And there are other factors that play into this too. You also learned about osmosis through the dialysis tubing experiment, where water diffuses over a membrane from areas of what? Higher concentration to lower concentration until equilibrium is produced, and that is osmosis. You learned about the two different types of cells, prokaryotic, which are bacterial, and eukaryotic, which is everything else, animals, plants, fungi, and protists. And you also learned about viruses. And if you watched my announcement video, you learned a little bit more, hopefully, about the coronavirus, SARS-CoV-2, which of course has been what everybody's been talking about for the last 2 years. So this week, what are we learning about? We're learning about cellular respiration. This is the production of ATP, and ATP is the energy molecule that runs all of the enzymes in cells. And enzymes carry out the biochemistry. They're the little helper molecules that make all of the chemical reactions happen. Well, they require energy, and they do that through the molecule ATP, adenosine triphosphate, and we get that from the breakdown of food and nutrients that we take in as organisms. We do this as aerobes, so aerobes means that we live in the presence of oxygen and use oxygen. We use oxygen to help us do this through a process called aerobic respiration. And respiration, you know, that's familiar to you, is like breathing in and breathing out. So why are we breathing in? Why are we breathing out? We're breathing in to take in air and collect all the oxygen from that air to do cellular respiration. And when we breathe out, we exhale carbon dioxide as a waste product. That is cellular respiration. There are a couple of other types. For example, there's fermentation, and there's lots of microbes and small organisms like types of fungi, yeast, etc., that can do fermentation. There's also a process called anaerobic respiration, but in fermentation and anaerobic respiration, no oxygen is produced. If you're a bigger organism, though, like us, like plants, you have to do aerobic respiration to gain enough ATP molecules from the food that you consume. We also touched this week on photosynthesis, and this is the production of glucose by using sunlight energy, putting together carbon dioxide molecules in water to make the sugar glucose. Now, we don't do much this week on that, but you do need to know about it, and if you're taking the lecture, you definitely will touch on it. All right, so let's see what we've got going on this week as far as lab goes. The lab this week, you must start by Thursday, and that's because you have to soak the peas that you get in your kit that are dried. You have to soak them overnight for 1 whole night, and then you have to put them on wet paper towels in the dark for another day. That's 2 whole days of working with the peas only. And then the third day, you set up the respirometers. So a respirometer is a little tube which is going to measure how much oxygen is being consumed in the air. So the little peas, hopefully, are going to do cellular respiration, and they're going to consume oxygen. Now, remember that we said even when we breathe in, we take in oxygen, we get the oxygen out of the air, but then when we breathe out, we exhale carbon dioxide. So there wouldn't be a way to measure this because the little peas would be taking in oxygen, but then they would be breathing out carbon dioxide. There wouldn't be any change in the gas volume in the respirometer. However, we are putting potassium hydroxide on some cotton inside the respirometer, and the potassium hydroxide will bind with and precipitate the carbon dioxide. So any movement in the gas volume in the pipette that you have will be an indication of how much oxygen is being used by the germinated peas, by the ungerminated peas, and by the plastic beads that you are going to be setting up in your respirometers. Just follow the directions. It's not that difficult to do, but it does take several days. You have to start by Thursday in order to get your lab report finished by Sunday. My suggestion is to start this even earlier in the week to make sure everything works. A couple of hints here. Potassium hydroxide is caustic. Use your safety equipment. Wear your glasses, wear your gloves, make sure nobody's in the room with you. Clean it up safely. You do not want to get this on your skin. You do not want anyone else to get this on their skin. So handle this very carefully, but it's necessary because it precipitates the carbon dioxide, and that way you can see movement of the gas in the respirometer. So a respirometer is just a little contraption or apparatus that measures what? Cellular respiration. That's all that means. Make sure you use your lab report forms. This is going to be very important. Very important. So use your lab report forms and combine everything into your one report to do your analysis. All that you have this week is this experiment. But like I said, it takes a little bit of time to do this because you have to prep at least 2 days before you can run the actual experiment, and then you have to write the lab report. I have put an extra video in the announcements on how to use parafilm because you have to do that this week. You are not familiar with parafilm. You've never used parafilm. It is one of the coolest things ever in the world. It is a stretchy type of wax paper, and it sticks to itself. So, my video is like a few seconds long, and it shows you how to grab onto the parafilm, twist it around back on itself, and it holds. So it's a very cool material to use. All right. And with that being said, I hope that you have a wonderful week. But if you need anything, just email me, m.sigmon@snhu.edu. I will be glad to help you in any way that I possibly can. So let me know. That being said, have a wonderful week number 4.